Bismillahirrahmanirrahim. Aslam alaikum to all of you. I'm Nazia Aslam, an MPhil scholar from Government College University, Faisalabad. I'm working under the supervision of Dr. Sara Zafar on the project Enhancement of Maize Tolerance to Salinity Stress through regulation of antioxidant defense mechanism. So here is our problem background. Food security is affecting about 1/3 of the total irrigated lands on Earth. Salinity is the most detrimental factor among the abiotic stress factors to economic agriculture yield over the world. Stresses trigger a wide range of the plant responses, just like cellular metabolism, altered gene expression, and changes in growth rates and crop yields. We were dealing with the maize plant. In Pakistan, maize is the third most important crop after wheat and rice. Being an important crop, maize is grown on about 1 million hectares with a total yield of about 2 million tons. It is also known as queen of cereals or golden crop. In view of its increasing importance, improvement in agronomic characteristics of maize plant has got considerable attention in Pakistan. It is used as food and corn oil for human consumption, feed for livestock and poultry, and raw material for agro-based industries. Maize contains about 10% protein, 5.8% fiber, 72% starch, 3% sugar, 5% vitamin A and B, 4.8% oil content, and about 1.7% ash. Lower yield of the maize has various reasons, just like salinity, wastewater fertigation, water scarcity, heavy metal pollution, poor soil nutrition. So what is salinity stress now? Soil salinity is the salt content in the soil. The ions responsible for salinizations are sodium, potassium, calcium, magnesium, and chloride. The salinity due to NaCl is the burning issue that highly reduces the plant yield, especially in the areas with limited rainfall, just like desert. It is estimated that about 1/3 of the irrigated land is affected by salt. Salinity has many socioeconomic effects on human life, low plant productivity, and economic returns. In our research work, we were dealing with the two hormones, just like melatonin and indole-3 acetic acid. Here is the production method of both. Tryptophan works as the precursor for both of these hormones. Tryptophan is converted into tryptamine, then to serotonin, and finally to melatonin. On the other side, tryptophan is converted into indole-3 pyruvic acid, indole-3 acetaldehyde, and then finally to indole-3 acetic acid. What are melatonin and indole-3 acetic acid now? Melatonin is a newly discovered hormone found in plants. Melatonin present in many plant parts, just like roots, seeds, leaves, and in flowers. The plant families, just like Brassicaceae, Rosaceae, Poaceae, and Apiaceae, contains larger concentration of melatonin. Its concentration ranging from picogram to microgram. Melatonin has oxygen-like effect on plants. Now, what is indole-3 acetic acid? It is basically a white color solid compound with molar mass of about 175.187 gram per mole. It is most commonly naturally occurring plant hormone of auxin class, predominantly produced in the cells of apex, just like bird, and very young leaves of the plant. Its biosynthesis starts from tryptophan, but also via other independent pathways. It inhibits the photorespiratory dependent cell death that suggests the role of indole acetic acid in stress tolerance. So here are our objectives. The first one, to elucidate the potential of exogenously applied melatonin and indole-3 acetic acid on growth, yield, physiological and biochemical traits of maize plants undergoing salinity stress. And the second one, to investigate the interactive effect of the melatonin and indole acetic acid application for improving salt tolerance in maize plant. Here is our plan of work. We were dealing with the two genotypes. First one was pearl and the second one was aqua. Salinity levels applied were 0 and 80 millimolar. Melatonin levels applied were 0 and 600 micromolar. Indole acetic acid level applies were 0 and 2 millimolar. Experimental design was CRD, and we were dealing with the 3 replicates. So here are our results. The first parameter observed was shoot fresh and dry weight. We observed under salinity stress, shoot fresh and dry weight decreased significantly, but with application of melatonin and indole acetic acid in combined form, shoot fresh and dry weight increased in a very high rate. as compared to the melatonin and indole acid when they are applied separately. The next parameter is basically the MDA content, which is an oxidative stress indicator. When we applied salinity, then there is a sharp increase in the MDA content. But with the application of melatonin and indole acid in combined form, there is a sharp decrease in the MDA content. The next parameter was the enzymatic antioxidants, which includes catalase, SLD, and peroxidase. All of these content were increased greatly when we applied salinity, but with the application of melatonin and indole acid, they observed a decrease in all these content, especially when we applied melatonin and indole acid in combined form rather than to apply in separate form. So here is our next parameter, including osmotic potential, water potential, and turgor potential. With increase in salinity, there observed an increase in the osmotic and water potential, but with the application of melatonin and indole acid, especially in combined form, decreases the osmotic and water potential, especially when we applied melatonin and indole acid in combined form as compared to their application separately. Next, the turgor potential decreases with the application of salinity, but increases sharply when we applied melatonin and indole acid, especially when we applied them in combined form, as compared to their application when we applied them separately. The next parameter include the observation of an enzymatic antioxidant, which is APX ascorbate peroxidase. When we applied salinity, then they observed a sharp increase in APX content, but with the application of melatonin and indole acid, they observed a decrease in the APX content, especially when we applied melatonin and indole acid in combined form rather than to apply in separately. Next parameter was the observance of H2O2, which is an oxidative stress indicator. The content of H2O2 increased with the application of salinity, but decreased when we applied melatonin and indole acid in combined form. The next was flavonoid, which is a non-enzymatic antioxidant. Its concentration increases when we applied salinity, but with the application of indole acid and melatonin in combined form, it decreases sharply. The next content was of proline, which is an osmolyte. It also increases by the application of salinity, but when we applied melatonin and indole acid in combined form, they observed a reduction in its quantity as compared to the application of melatonin and indole acid when we applied them separately. The next parameter was to observe the gas exchange attributes, which includes the photosynthetic rate, transpiration rate, and stomatal conductance. Under salinity stress, the photosynthetic rate is decreased along with the transpiration and stomatal conductance rate. When we applied melatonin and indole acid separately, then they observed a little bit increase in these gas exchange attributes, but the significant change was observed when we applied melatonin and indole acid in combined form. The next parameter was to observe the nutrient contents, which includes nitrogen, zinc, iron, and calcium. Under salinity stress, all these contents were reduced significantly. When we applied melatonin and indole acid separately, they observed a little bit increase in these nutrients, but the significant change was observed when we applied melatonin and indole acid in combined form. They observed a very sharp increase in all of these contents. The next parameter was to deal with grain weight and yield of the plant. Under salinity stress, both of these were reduced significantly, but with the application of melatonin and indole acid, their contents or quantity increases. But the sharp increase was observed when we applied melatonin and indole acid in combined form, rather than we applied melatonin and indole acid separately. So here is our conclusion. Oxidative stress indicators increase in stress and decrease with application of melatonin plus indole acid. A considerable decrease in growth, photosynthetic attributes, nutrient analysis like potassium, zinc, iron, and yield attributes were observed under saline environment. However, the application of 600 micromolar melanin plus 2 millimolar indole acetic acid foliar application is the most suitable in regulating growth of maize plants under saline environment. Thank you all.